Good afternoon, students. Today we are here in front of you to discuss the biochemistry portion of Grand Test 180. Now let's see question number 1. Which of the following is not a protein misfolding disorder? Now see, protein misfolding disorder, all of you know that synthesis of proteins takes place in our body. The three major organelles which play a vital role in the synthesis of proteins are Golgi apparatus, ribosomes, and the endoplasmic reticulum by a process known as signal transduction. Once the proteins are formed, they undergo again an editing process whether there is misfolding protein or not, and this is corrected by chaperones, which are basically the heat shock proteins which help in the or which prevent misfolding of these proteins. And they utilizes ATP for their action, and that is why they are also known as heat shock proteins. Now here the question is ki what are the major disorders which are due to the protein misfolding? Obviously the answer is tuberculosis. Baki rest of the diseases are linked by proteins, so they are due to the protein folding disorder. Jaise CFTR is there, beta amyloid protein are there, which are basically responsible for this. So here the answer is clear, it's tuberculosis. Then question number 2. Now see, before starting up with this, we should know what do we mean by this gluconeogenesis process. This is very, very important, and every year a question is asked on gluconeogenesis. Now, as it is clear from the word itself, gluconeogenesis, synthesis of glucose, from carbohydrate precursors and from non-carbohydrate precursors. Now carbohydrate precursors means it is strictly a reversible process of glycolysis, or you can say the 3 steps of glycolysis, irreversible steps. They are made reversible in gluconeogenesis. Now see, gluconeogenesis is the most important process which takes place during starvation, and it is an anabolic process because it leads to the synthesis of glucose. This is a very important question. This has been asked in the exam. Now see, the last step of glycolysis is conversion of PEP to pyruvic acid. So here in gluconeogenesis, this step, conversion of pyruvic acid to PEP, will become the first step. Now conversion of PEP to pyruvic acid is a single step reaction, but in gluconeogenesis, conversion of pyruvic acid to PEP takes place in 2 steps. Now what happens is, you can see this pyruvic acid is present in the mitochondria, where it simply forms acetyl coenzyme A by oxidative decarboxylation, and this acetyl coenzyme A is combining with oxaloacetic acid to form citric acid, and this is how the citric acid cycle goes. Now what will happen is that this pyruvic acid will be first of all converted into oxaloacetic acid in the presence of enzyme. See, pyruvic acid has 3 carbon atoms, oxaloacetic acid has 4 carbon atoms. Simple, there is addition of 1 carbon dioxide. Carbon dioxide, so you can say enzyme is pyruvate carboxylase. And this pyruvate carboxylase requires ATP for its function, first thing, and second is it requires biotin as a coenzyme. Wherever carboxylation occurs, biotin is required. Now what will happen is that this pyruvate carboxylase is modified, allosterically modulated by this acetyl coenzyme A. Basically what happens is that this acetyl coenzyme A, the production of acetyl coenzyme A will increase due to the beta oxidation of fatty acids. During starvation, what will happen? Beta oxidation, more the rate of oxidation of fatty acid, more will be the acetyl coenzyme A formation, and more it will activate this gluconeogenesis. So you can say that acetyl coenzyme A, it is an allosteric modulator, this you have to remember. And it accelerates, it modulates the activity of pyruvate carboxylase. Now see, what happens is, this gluconeogenesis is under regulation by so many hormones. सबसे important, the most important hormone which plays a major role in this is glucagon. This glucagon activates gluconeogenesis. First is obviously it is going to accelerate the activity of this acetyl coenzyme A, one factor. Second is this glucagon acts on pyruvate kinase. Pyruvate kinase, the enzyme involved of basically, see what is happening is if glycolysis is occurring, there is no question of gluconeogenesis and vice versa. So all the irreversible step of glycolysis, all the you can say regulatory enzymes of glycolysis have to be made inactive during gluconeogenesis and vice versa. So gluconeogenesis mein all those enzymes will be activated which are inactive in glycolysis. Jaise pyruvate kinase is a very important enzyme of glycolysis, which is an example of substrate level phosphorylation also, in which this PEP is converted into pyruvic acid. So it is present in 2 forms, active form and inactive form. Glucagon always increases the concentration of cyclic AMP, and the function of this cyclic AMP is, the function of this cyclic AMP is simple, making protein kinase active. If it is making protein kinase active, on the other hand, obviously protein kinase, here it is pyruvate kinase will be inactive. Or you can say the inactive form of pyruvate kinase is phosphorylated. This is a very important question. So glucagon is basically accelerating the conversion of protein kinase from inactive to active form. And when pyruvate kinase, when protein kinase is made active, obviously pyruvate kinase will be made inactive, or in other words we can say inactive form of pyruvate kinase is phosphorylated, which is accelerated by glucagon. So glucagon is basically acting on gluconeogenesis by 2 forms. One is it is accelerating the activity of acetyl coenzyme A, which is an allosteric modulator of this enzyme, and second is it is making the pyruvate kinase inactive. And same is the case here. If you see the question is which of the following is most effective acetyl coenzyme stimulation of pyruvate carboxylase. Pyruvate carboxylase enzyme, first enzyme of gluconeogenesis, which converts pyruvic acid to oxaloacetic acid. And one very important thing about this reaction is this is one of the most important anaplerotic reaction or the filling up reaction. Whenever there is increased flux of pyruvic acid, it starts increasing the production of oxaloacetic acid. Now this oxaloacetic acid will finally be converted into PEP by taking malate dehydrogenase as one of the very important enzyme. Because oxaloacetic acid is impermeable to the mitochondrial membrane. It is converted to malic acid. Malic acid is transported from the mitochondria to the cytoplasm, and then again it is reconverted back to oxaloacetic acid. And this oxaloacetic acid will finally be converted into PEP by this PEP carboxy kinase enzyme. Basically this gluconeogenesis, this is again a very important question. It always takes place in the cytosol, but it involves mitochondria also, because the first anaplerotic, first reaction of gluconeogenesis takes place in the mitochondria. Then question number 3. Glycogen synthesis and breakdown takes place in the same cell having enzymes. This freshly synthesized, and it is not because glucose 6-phosphatase is present on the endoplasmic reticulum and cannot act on the obviously glycogen present in the cytoplasm. Every enzyme has its own site. It is present in the endoplasmic reticulum itself. Now here what you should remember in glycogenesis and glycogenolysis is these two are the antagonistic processes. Glycogenesis and glycogenolysis are the antagonistic processes. Genesis and glycogenolysis. You should always remember the rate limiting steps. Rate limiting step in glycogenesis is glycogen synthase. In glycogenolysis it is glycogen phosphorylase. And this glycogenesis, simple, it is formation of glycogen from glucose, lysis breakdown of glycogen to glucose. The last enzyme here is which converts glucose 6-phosphate to glucose. It is glucose 6-phosphatase, which is actually absent in the muscle. And that is why glycogenolysis never contributes to blood glucose directly in the muscle. And that is why their Cori cycle is operative. Whereas in liver, obviously it takes place in the liver itself where glucose 6-phosphatase is present. And Cori cycle is a cycle which is involving both the organs, liver and the muscle, where reuse and resynthesis of glucose is there by taking lactic acid is one of the major intermediates. So obviously here the answer would be glucose 6-phosphatase is present in the ER and cannot act on the glycogen which is formed in the cytoplasm. Is it clear? Then question number, what is the code for selenocysteine? Now see, ideally what we have studied is, we have studied that there are 20 standard amino acids. 20 standard amino acids. But after the discovery of these two new amino acids, they are known as 21st and 22nd amino acids. 21st is selenocysteine, which is coded by UGA, and pyrrolysine, which is coded by UAG. Both are important. The only disadvantage is that they are coded by the nonsense codons. And the selenocysteine, as it is clear from the word itself, here what is happening is selenium is required for the synthesis of this selenocysteine, which has cysteine as one of the major intermediates. And this selenocysteine is a very, very important enzyme of so many other enzymes, जैसे thioredoxin reductase. Thioredoxin reductase. Similarly, glutathione peroxidase. Glutathione peroxidase. So here it is what? Ki simple, you can say ki selenium and cysteine are required to synthesize selenocysteine. The enzyme here is selenocysteine synthetase. Synthetase. Why synthetase? Because it cannot occur without the use of ATP. You know what is the difference between synthase and synthetase? Synthase is simple synthesis of the compound is there without involving ATP, and synthetase is whenever ATP is involved. This is again a very important question. Selenocysteine synthetase, coding या forming selenocysteine coded by UGA, which is the 21st amino acid. There are certain derived amino acids also. Derived amino acids means which are derived from these. And these derived amino acids include ornithine and citrulline. Right? So the answer here is selenocysteine, which is coded by UGA. The next question, whenever we talk about, you have to first of all see what is the question like. Which of the following conversions does not require biotin as a cofactor? Basically, biotin is a coenzyme. Coenzyme means, you see every enzyme has a prosthetic group associated to it. Prosthetic group associated to this. If this prosthetic group is a metal ion, it becomes a cofactor. अच्छा ठीक है नहीं ये है शायद 5th क्वेश्चन Answer to question number 5th. C. Cofactor. You know every enzyme is a protein, and this enzyme cannot work alone. It requires a prosthetic group to work along with it. Prosthetic group. If this prosthetic group is a metal ion, it becomes a cofactor. If it is an organic molecule, it is a coenzyme. Always remember all the vitamin B complex vitamins they come under the category of coenzymes. And this is again a very important question. Whenever we talk about carboxylation, for carboxylation, biotin is required, always. Biotin is a coenzyme for carboxylation. Similarly, PLP is a coenzyme for transamination and decarboxylation. Now, whether it is acetyl coenzyme carboxylase, this is an enzyme, first rate limiting enzyme of fatty acid synthesis. It requires biotin. Only vitamin K, which is also carboxylating, the glutamic acid residues, in prothrombin does not require biotin, because this is basically the property of this vitamin K, which is a fat soluble vitamin and which helps in the carboxylation of glutamic acid residues which are present on the sides of this prothrombin by taking epoxide as one of the major intermediates. Epoxide reductase is the name of the enzyme also, which is basically inhibited by the action of rat poison and dicumarol. So this epoxide reductase, formation of epoxide being an intermediate, no requirement of biotin is there, and rest of the examples you can see in the question itself. Gamma carboxylation of glutamic acid, this is what is the action of vitamin K. Acetyl coenzyme A to malonyl-CoA, first rate limiting enzyme of fatty acid synthesis, enzyme required is acetyl coenzyme A carboxylase. Similarly, propionyl-CoA to methyl malonyl-CoA. Propionyl-CoA, this is one of the reactions of gluconeogenesis, conversion of this to this. Here the enzyme is propionyl-CoA carboxylase, which again requires biotin. And pyruvate to oxaloacetate, just now we discussed this reaction, it requires biotin for its action. So it's clear, so the answer is gamma carboxylation of glutamic acid. When a 10-month-old child, now see, the answer to this question is eye cell disease. Because basically what happens is, this eye cell disease is again what happens in this disease is there is glycosylation of mannose forming mannose 6-phosphate. The site of this glycosylation is basically on this is basically N-linked linkage. N-linked linkage in the Golgi apparatus. And this is a type of mucolipidosis only. Mucolipidosis. So isme here again we can do the, because if you see the question, it is somewhat similar to Hurler syndrome also, but major difference is that this enzyme is deficient. So this is why the answer to that question is the eye cells disease. Basically, see, if we talk about glycosylation is nothing but attachment of carbohydrate residues to form either proteoglycans or glycoproteins. Now, whether it is a N-glycosidic linkage, whether it is a O-glycosidic linkage, that depends upon the linkage. If we call it as a N-glycosidic linkage, it means the proteins, the carbohydrate is attached to that protein which is rich in the basic amino acids, lysine and arginine. Similarly, if it is O-glycosidic linkage means that they are rich or they are connected to either the amino acids which are rich in hydroxyl group containing. Now, next option is Hunter syndrome. Hunter syndrome is basically due to, the option next is MPS type 2. MPS type 2 is nothing but Hunter syndrome, which is due to the deficiency of L-iodouronate sulfatase. C is Proteose syndrome. This Proteose syndrome is nothing but it is basically due to the malfunctioning of glypican 3 gene. I'll write it. Glypican Protease is due to glypican 3, and then Larsen is due to filamin B. Filamin B, which leads to different cytoskeletal disorders. So the answer here is eye cells disease, as the enzyme is deficient, and basically it is what it is helping in glycosylation of that particular polysaccharide, right? The next question, specific activity. Now, what do you mean by the term specific activity of the enzymes? See how active an enzyme is. It means कि how active an enzyme is, कि this enzyme is reacting with the enzyme substrate complex. Enzyme is reacting with the first of all substrate to form this enzyme substrate complex, which dissociates to form the free enzyme and the substrate. So the entire kinetics of the enzyme is based on this. So अब suppose if I say कि how active an enzyme is means कि there is a rate of conversion कि 1 micromole of a substrate can be converted into a second. That will be the activity of the enzyme. Or it is also referred to as Kcat also. Kcat is basically the catalytic constant, which is higher for carbonic anhydrase. And second is trypsin. So an international unit of this enzyme activity is kettle. So here you can see what are the options. Options are micromoles per minute, milligram per micromole. Simple, if we classify it enzyme activity in terms of measurement, it has to be unit of activity per milligram of that protein. And internationally it is known as 1 kettle. Then question number 8. The pH of body fluids is stabilized by. अच्छा वेट ठीक है. Next question number 8. Question number 8 का answer would be obviously when pKa value is associated to 7.21 and these acts as the best buffers. The reason being is कि they are basically touching the physiological pH. Physiological pH. So it is pKa 7.21. Then next question. The following test is performed for which condition? Now see here you can see it is simply hereditary spherocytes. Hereditary spherocytes mein you can see ki whenever we compare this type of test, one is a control sample and other is the test sample. Control mein you can see a line, whereas in the test sample that is invisible. Why? Because there is excessive hemolysis of the RBCs and that is what exactly happens in spherocytes. So the answer of this is A. And L-captonuria, all of you know, it is basically due to the deficiency of homogenistic acid oxidase where the urine turns black on exposure to air. And when we compare all the 4 options, the test here it is spherocytosis for this reaction. Then next, during evaluation of a mentally retarded child, following findings were noted. अब यहाँ पे it's clear it is alpha L iodouronidase enzyme, which is one of the very important mucopolysaccharidosis. So the answer here would be A. Now alpha L iodouronidase deficiency leads to which disease? It is Hurler's syndrome. Hurler syndrome is due to the deficiency of alpha L iodouronidase. There are basically 7 types of mucopolysaccharidosis: Hurler syndrome, Hunter syndrome, Ski syndrome, Martwick syndrome, Ski syndrome and Sly syndrome. Then next question. The pentose phosphate cycle produces which of the following sugars? Now see, this is again a very important question. Pentose phosphate, basically you should know this is known as the shunt and it is known as hexose monophosphate shunt. It is a multicyclic process. The significance is it generates NADPH and it generates ribose sugar. NADPH, which is required for the synthesis of fatty acids, for the synthesis of cholesterol, for sphingolipidosis, for sphingolipids. NADPH is required. And ribose sugar, which is a precursor of purines. As all of you know that these purines are synthesized by a de novo pathway, and this ribose, which is coming from HMP Shunt, it is the precursor of adenosine monophosphate and guanosine monophosphate. So the answer here is ribose sugar. Question number 12, vitamin B1 deficiency. Now this is again a very important question. It is generally asked in the exam. You should know this vitamin. The active form of vitamin B1 is thymine. Active form of thymine is TPP. And this is one of the very important coenzyme for pyruvate dehydrogenase complex. This is an example of multi-enzyme complex. And it is required for the conversion of pyruvic acid to acetyl coenzyme A. Simple decarboxylation occurs. and this requires TPP, lipoic acid, FAD and NAD for its activity. And it requires E1, E2 and E3 other enzymes. Now simple question is, obviously when this TPP is deficient, because this is the major carrier of this carbon dioxide. If this is absent, that means no formation of acetyl coenzyme A will be there, and this pyruvic acid will be converted to lactic acid, or there will be lactic acidosis due to the deficiency of TPP, which is the major constituent of pyruvate dehydrogenase, which is an example of multi-enzyme complex. So the answer here is pyruvate dehydrogenase. The next question, which of the following trace elements is required for conversion of procollagen to collagen? For this, copper is required, and this reaction requires this. Basically, collagen is synthesized from procollagen, and there is, so number 13. Collagen consists of 3 repeats, triple repeats होते हैं इसमें, lysine, proline and glycine. Lysine is hydroxylated, proline is hydroxylated to form hydroxylproline and hydroxyl lysine, and this glycine is simply glycine. Now this hydroxylation requires vitamin C for its function. Means vitamin C is required for the hydroxylation of lysine and proline, and the metal involved is copper in this case. Because or you can understand it like this, कि for hydroxylation copper is the basically element required to hydroxylate different residues. So the answer here is simple copper. And a very important thing is deficiency. That is why deficiency of vitamin C leads to defective collagen formation. Collagen formation. Or it leads to scurvy. So synthesis of collagen is very, very important. Then question number 14. Regarding HMP, which is a fructose. Now see, fructones are simple example of fructones. It is clear from the word itself. Fructones are nothing but the polymers of fructose. And this comes under class homopolysaccharides. Actually, homopolysaccharides are the, you can say, which have same identical units of monosaccharides. So, starch, glycogen, dextrins and cellulose, they all are glucogens, as they require glucose, whereas inulins are the only fructogens which are present, and their main function is that they help in the estimation of GFR. So fructosen is only inulin, and rest of them are glycogens or glucosens, as they consist of the identical unit of glucose. Then question number 15. Regarding HMP shunt, see, first of all you should know there is a difference between HMP shunt and glycolysis. Glycolysis is also known as EMP, Embedon-Meyerhoff-Pranav's pathway. It is not a shunt. HMP is a shunt, hexose monophosphate, which never forms an end product. One thing, it is a multicyclic process. No ATP is produced in HMP, whereas ATP is produced in glycolysis. And then CO2 is produced in HMP, not in glycolysis. So here it is, occurs in the cytosol, yes. No ATP is produced, it's active in adipose tissue, yes. Liver plays a very important role, and the oxidative phase generates NADPH, and non-oxidative phase. The answer to this question is D. It never forms pyruvate. Never forms pyruvate. Simple, it starts with 3 molecules of glucose 6-phosphate, 6 NADP plus 6 H positive, giving rise to 2 molecules of glucose 6-phosphate plus 6 NADPH plus 3 carbon dioxide plus glycerolaldehyde 3-phosphate. This is the entire equation of HMP shunt. So, non-oxidative phosphate, the non-oxidative phase never forms pyruvate. Then last question. Regarding proteoglycans, false is. Chondroitin sulfate is a proteoglycan. They hold less amount of water. Yes, they do not hold less amount of water. They hold large quantity of water. As it is clear from the word itself, proteoglycans. Proteoglycan means carbohydrate is present in bulk. Actually, there are 2 types of proteins, proteoglycans and glycoproteins. The only difference is when it is whatever is the suffix, it has to be present in more amount. In proteoglycans, carbohydrate content is more, which is soluble in water. In glycoproteins, proteins are there, that depends on the charge, या which amino acids are present in it. So solubility will depend on that. So out of these 2, if we have to compare solubility, then proteoglycans are more soluble. So the answer to this question is B. So here we finish up with the discussion part of the biochemistry. Thank you so much.